All right, moving on to our last speaker of the day. It is Lloyd Jeffs, who is the Director of Clinical Manufacturing Solutions here at Precision NanoSystems. Lloyd's going to talk about PNI's biopharma services and how we help our clients along the path to genetic medicine development and manufacturing. So thanks, Lloyd, and take it away.
All right, thank you for the introduction, Josh. Hello, everybody. So yeah, I'm going to be talking about how Precision NanoSystems helps our clients and customers with biopharma, with our biopharma services group and team. So we have basically a complete range of turnkey solutions for accelerating and de-risking the development of genetic medicines. So Precision NanoSystems also develops other nanomedicines technologies, and they can't be ignored. They're very important, especially for delivering small molecule drugs. And we look at other delivery systems as well, in addition to lipid nanoparticles, where we're looking at polymeric solutions, emulsions, and actually lipopolymeric solutions as well. So this talk, just as an example, is going to focus on lipid nanoparticles for the delivery of RNA and DNA. But mostly RNA delivery using lipid nanoparticles and the services that we can provide to support that. Okay. So PNI started with our instruments and systems, our microfluidic-based instruments and systems. And we have basically, in order to help our clients more, we've provided or started about 5 years ago to provide services. So we have many different customers at many different stages of the drug development process. So the earliest stage would be discovery and development, and also moving then on to early preclinical development. And those customers may already have one of our systems, one of our nanoassembler systems, but need help with their formulation development or need to accelerate their process development activities. And we can offer that. And I think that's a key advantage. So we can help our customers with basically whatever they need in their drug development process. So, sort of if they need help with a specific area, we can do that. But everything's centered around our microfluidics technology. And we think that's a world-leading technology. We know it is. Our clients are telling us that as well. So, the power of our technology with the services that we offer, and also not to forget our reagents, our clinical lipids, and our basically R&D, RUO reagents such as Genvoy, are really helping our clients move quickly in this area. Because time is of the essence, as we all know, especially in dealing with the COVID-19 pandemic. And the faster we can provide those services and help move our clients forward, the greater the success we're going to have, right? So in later stage preclinical development, our systems are the Blaze that can make up to 10 liters of product, and our GMP system, which can currently make up to 50 liters of product and can support and is supporting phase 1 clinical trials. So back to our services. So I'm going to talk about basically how you go from the beginning in a case where a client comes to us and wants screening all the way through to technology transfer and GMP manufacturing. So the platforms we offer in the center of this slide are now, and recently we are about to offer basically genetic payload platform, which comprises of the design and manufacture of self-amplifying messenger RNA, or SAM, or saRNA, as it's sometimes known or referred to. And we can offer that as a service. We can offer a delivery platform, the Genvoy delivery platform, with PNI's proprietary lipids and formulations. And combined with our nanoassembler microfluidic manufacturing platform, we can really move forward projects very, very quickly. We can provide all the pieces. An important part of that is the people providing the services. So we have a lot of expertise here at PNI, concentrated expertise. collaboration within the team and also close collaboration with our clients is essential for success, and we've realized that. So I'm going to talk a little bit about our team and our expertise in the next couple of slides. So I'm very proud of the team that we're developing here at Precision NanoSystems. Our team spans from chemists to analytical scientists to formulators, biochemists, molecular biologists, and not to forget our quality team as well that oversees our later stage projects. We have in our biopharma services team, we have 19 full-time employees with advanced degrees. We have a lot of experience that we either generated here at PNI or brought from other work experiences prior to joining, where teams have worked together in total on over 26 clinical stage programs, made greater than 44 GMP batches, and some of our team have been involved in producing or manufacturing or support of drug approvals as well. So we have experience across a broad range of drug payloads and carriers, not just RNA and lipid nanoparticles, small molecule drugs and other types of nanomedicine delivery systems. And right now at PNI, we've either completed or is in progress 154 client projects, which I think is quite significant and quite impressive. For each of those client projects, we do maintain confidentiality. So we do build close trust and collaboration with each of our clients, which is of course essential. So hopefully you're getting the idea that with our integrated suite of custom services and our platform microfluidics technology, we can really have all we need to help our clients accelerate and move as quickly as possible toward the clinic. And we can cover the entire drug development continuum, obviously from proof of concept all the way through to GMP production. And PNI can use its NanoSampler platform to develop optimized process parameters while keeping in mind these critical quality attributes that sometimes the client brings with them or we suggest to the client. And we are using the same systems and tools that we're offering to our clients. So I think the power of that technology is that we can develop a process using an Ignite instrument, for instance, using a GenVoid reagent, and using our saRNA payload that's all developed at PNI. We can transfer that to a client on another side of the world, and they can generate basically the same quality particles with the same physicochemical properties and the same biological activity as we can in our lab. So that transfer of knowledge, technology, methods, procedures is seamless, and we ensure that that is seamless. And I don't want to forget this, but any successful project has a well-defined project plan. So our project management team here at PNI will ensure the objectives and the scope of that plan are clearly stated at the beginning. We'll track progress, make any course corrections or changes that are required to the plan, but ensure that the client basically gets what they need, gets the deliverables within the timeline and the budget that's been agreed to. So we have a comprehensive offering of custom formulation and manufacturing services that I've mentioned. And this is just some examples here on this slide, all the way from discovery and commercial and how we help customers. With the screening, we are using the Spark system to screen nanoparticles to identify hits and further development for clients. We're also using the Ignite system, which replaced the benchtop system, our original system a couple of years ago, to develop and optimize nanoparticle physicochemical properties and biological activity. And at this point, we're identifying or confirming the critical quality attributes of the particular nanomedicine, whether that's a therapeutic or a vaccine, and thinking about drug product specifications, right? What is the ideal particle size? What is the lipid to drug ratio, or the ratio of your payload, your RNA, for instance, with that of your lipid carrier? And at this point, our analytical team gets involved and talking with the client, seeing what analytical methods are available that have already been developed, and also assessing what methods need to be adapted or created to properly understand and characterize the particular nanomedicine that's being developed. In the preclinical realm, we are using the larger Blaze system to help us do process development, to make larger batches to help develop and establish process parameters, including things like hold time. One of the challenges, as many of you already know, with RNA-based vaccines and therapeutics, the RNA itself, especially longer RNAs like messenger RNA and self-amplifying RNA, are somewhat labile. So you have to be very careful with the conditions that you use to manufacture the drug and the conditions you use to choose to store the drug as well. And most of those drugs are currently stored under frozen conditions, including the Moderna and Pfizer-BioNTech COVID-19 vaccines. In addition to the process development for the particle formation step, we're also experts in optimizing downstream processing steps, such as tangential flow filtration, and also sterile filtration of drugs as well. So once the process parameters have been identified and set, and ranges have been set, we would then move to our GMP nanoassembler system for the scale up to the scale that's required to manufacture GLP tox and ultimately GMP batches. And we've seen for a number of projects now that you get a very predictable translation from the different systems, the Ignite to the Blaze to the GMP system of the physical chemical properties. That's only part of the story though, right? So you can make the same particle as you measure it by particle size or the distribution or polydispersity of the particle, the amount of, or the proportion of the drug that's encapsulated or the encapsulation efficiency. But I want you to keep in mind biological readouts or bioassay is very important for RNA-based drugs. So we need to ensure that the biological activity is maintained throughout the manufacturing process and when you go from small scale into intermediate scale to your clinical scale. So those checks have to be performed all along the way, and especially if changes are made to the process or formulation during development. So, I want you to keep that front and center in your mind, that having a reliable and qualified in vitro bioassay is really going to help accelerate and ensure a successful project with an RNA-based LNP, whether it's a vaccine or a therapeutic. So moving on from scale up, once we've scaled the process here at PNI, or we've helped a client scale it at their facility, a technology transfer is required to a contract manufacturing facility or a GMP facility. Some of our larger clients have their own GMP manufacturing facilities. So that technology transfer will include the transfer not only of the equipment and the materials, but also of all the analytical methods and the manufacturing instructions for the drug product, along with all the quality documentation. So when we transfer an analytical method, it will be qualified. It will be in the form of an SOP that our analytical development team and our QC teams will work together on. Once the equipment and the methods are on site, we can assist with qualification at site of the analytical methods. We can assist with water runs and also engineering batches at the GMP site. And once those qualification activities and engineering batches are complete, we can then help with finalizing the GMP master batch record in advance of the manufacture of clinical batches. During manufacture, we can provide on-site or virtual support. It's basically what the customer or the client requires. We can provide that as a service, and we can provide any kind of CMC support during the drafting of any IND or clinical trial application. And that basically, we're able to do that because of the documentation that we provide for our systems, for our materials, and also for the work, the qualification work that we've been doing in process development and analytical and QC. Okay, so this slide is a summary of a path from a program kickoff with a client working with PNI, where the target product profile is discussed and presented by the client, such as what is the disease the drug is going to target, what is the desired concentration of the drug, what is the desired storage and shelf life of the drug to be developed. Just examples of that. What is the desired particle size? That can all be part of the target product profile. We'll work with the client to discuss and modify and fine-tune that, and then come up with a plan to achieve the target product profile as laid out by the client. So to reiterate, PNI, we're not a drug development company. We exist to help democratize and provide services to academia and to companies and government organizations that want and need to develop nanomedicine-based drugs, right? So we're here to provide a full suite of services. That's our mission. That's why we exist as a company. So once the target product profile has been established, PNI will set to work on nanoparticle formulation and process development work. Again, previously agreed to with the client. We'll also start with the analytical method development as soon as we can. And part of that analytical method development will be developing biological assays, such as an in vitro potency assay, to see how the drug performs after it's produced. While that's going on, we'll be looking at the particular, say, lipid formulation and the inputs for particle formation, and see if we need to configure or reconfigure our GMP microfluidic system. What I mean by that is in some cases, the lipid solution or the lipid mixture provided by the client will need heating. So we'll need to consider how we heat the lipid solution as part of our GMP microfluidic system. We've actually done that, and we can now offer that as a customized system. There are other considerations and requests that clients require, and we can customize our existing systems to do that. So once we've defined the formulation and locked the process, we will then basically take the R&D batches, continue on with stability studies for these early batches to give us an early look at how the particle, basically the product, the nanoparticles, perform with time. You know, what is the preferred storage condition? Obviously, you'd like to store your final product at ambient temperatures or 2 to 8, but we're seeing with RNA-based nanomedicines, they typically need to be stored under frozen conditions, either at -20 or at -80. So those initial stability studies will generate the data to make these data-driven decisions on drug development. Again, we will also, once we've locked that formulation, we'll actively start technology transfer to a CDMO. And the recommendation from us is that that work starts as soon as possible to basically identify a CDMO, get all the quality agreements, master services agreements in place, so that when we're ready, we can move quickly. That technology transfer process typically can take between 3 and 6 months. So it can be drawn out. We've looked at ways of accelerating that, but planning is the best way, or proper planning is the best way to accelerate any timeline. So start early, consider where you're going to make the GMP batch, and make plans to do that. Get the agreements in place, get your slots reserved. So after the technology transfer is completed, the engineering batch can then be performed at the CDMO or the GMP facility. And as I've mentioned, once the engineering batch has been basically analyzed, it'll be basically a batch, hopefully at the same scale you want to make your GMP batch, with the same quality of materials, with the same equipment. And you then have a chance to make some final tweaks to your master batch record and any tweaks you need to your analytical methods. And then that's locked for GMP batch manufacture. And, you know, we'll see then after the batch is produced, we'll have hopefully a GMP batch release, and all the data that went into the development of this product and all the production documentation and analytical documentation will be available to support a regulatory submission. So I'm going to spend a little bit of time talking about analytical development. I know this has already been covered in the webinar, and Adam Crow, who's our manager of analytical development, working with Derek Stebbing, our manager of QC, play a very key role here at PNI. And we have recently really expanded our analytical development and QC capabilities because it's so important. And we will continue to do that as new technologies become available. We will bring those into PNI. I think one of the things we want to do is stay on the cutting edge of the analytical, of our analytics, especially as it relates to RNA and the quality of RNA. So here are some examples of the critical components of an analytical development that are really, underpin the success or will underpin the success of a program. So it's not just good enough to make some lipid nanoparticles and say, hey, these look good. We need to understand and accurately measure the different properties of those particles and, importantly, the drug substance. So it's all about basically initially is our payload design, and we are offering that as a service as mentioned for our in-house self-amplifying RNA production. And we need good analytics to determine how pure that material is. Is it the right material? Is there any kind of degradation of those payloads that we need to be aware of, whether that's chemical or enzymatic? As far as the lipid nanoparticle composition goes, we need accurate methods to determine that we have the correct lipid raw materials. So they're shipped or made somewhere else, brought into PNI. We don't need any mix-ups, but also, very importantly, we need to accurately measure the relative compositions of each of those lipid components. And typically, an RNA lipid nanoparticle may contain 4, or in some cases, 5 different lipid components. So methods needed to be developed to measure each of those lipid components accurately and reproducibly. RNA quality, not only as it exists as a drug substance or an API, but in the formulated particle is also very, very important. And lastly, ensuring that we are maximizing the encapsulation of our RNA payloads is critical as well. But at the end of the day, in vitro and in some cases in vitro potency assays basically are telling you if your RNA is working, is functional in its final form. And it will tell you how long it is functional upon storage as well. So what we've done for different clients is we've assessed the in vitro potency of RNA LNP formulations during manufacturing and after manufacturing on stability as well. So, these analytical methods not only help with assessing the quality of the final product, but also inform us in our process development efforts as well. This is a list of some of the analytical capabilities of PNI, whether it's in the characterization of particles, where we use dynamic light scattering. We use regularly cryo-TEM. We're looking at kind of other approaches as well, evaluating for particle size characterization, and then standard things like, endotoxin, bioburden, pH, osmolality are also routinely performed as services by PNI. And some of those right now are being contracted out to third party, but I think our long-term goal is to bring in all of those analytical capabilities, including bioburden and endotoxin analysis. For the lipids themselves, we rely heavily upon chromatographic techniques, and we have in-house LCMS system when we have UPLCs with different detectors, UV, LSD, and CAD. For the payload, the genetic payload, in this case RNA, again LCMS, different UPLC approaches. We have a capillary electrophoresis system that we've been setting up from SciEx. And also, agarose gel electrophoresis techniques. And we use, for the RNA encapsulation, we use fluorescent dyes and the plate reader-based methods. And lastly, for bioassays, we are set up basically to develop and also routinely perform in vitro bioassays. So we have everything we need to do that here, including gel imaging, cell imaging techniques as well. So as mentioned, the analytical and QC services at PNI basically span the whole basically drug development process, from discovery and formulation screening. You need these methods, these analytical methods, to properly assess API and lipid stability. A lot of times in drug development, as you may know, the sooner you can actually down select, you can have a bunch of different candidate APIs and lipids. If you can find a way to select the good ones and maybe set aside the less optimal lipids and APIs, the sooner you can do that, the easier it's going to be. The sooner you can get to your lead, the better it is. And analytics drive that decision-making process. Early preclinical formulation and process development, having an HPLC or a UPLC-based method that allows you to measure each of the lipid components in a single assay is very powerful and will help accelerate development. Having, again, further developing methods to look at your active pharmaceutical ingredient is also critical. And basically having these methods that are robust and reproducible to give reproducible results, I think that is key as well. I think can be confusing sometimes if you have not properly developed your analytical methods and you're getting variable results and you need to make decisions on those results. That's going to slow down development significantly. So you've got to get your analytics right. And then, obviously, continuous improvement and optimization of assays to improve their repeatability, robustness is key as well. So we spend a lot of time in that zone, especially as we move analytical methods to quality control methods. And I think with our expertise in analytics and formulation, we're able to provide a pretty deep assessment of the particle physical characteristics, not only size and polydispersity, but also looking at concentration, density, the structure of the resultant nanoparticles, encapsulation efficiency, etc. As we move into late preclinical manufacturing, we are taking those analytical methods, we are beginning to qualify those, and our QC team's beginning to write SOPs that will be approved and then can be transferred to our clients' manufacturing facilities. Also, we'll be doing a lot of release and stability testing. So as many of you know, it might only take 2 or 3 days to actually make a GMP batch of an RNA LNP therapeutic. And the clinical manufacturing team can do that. They can actually get it vialed and stored. Then, for the next probably 2 years, that batch becomes the responsibility of our quality control group. They'll perform the release testing and they'll be performing the stability testing for, in some cases, greater than 24 months. So, it's basically the clinical manufacturing team will make the baby and the QC team will basically look after it, maintain it, monitor it, keep it safe for the remaining 2 years, for the rest of its life of the drug. So, the QC team deserves a lot of credit. It's not just the people manufacturing the drug, it's the people releasing it and testing it. And then the overlay there is the QA team that we have will be reviewing all the documentation, all the protocols before, during, and after completion of those protocols and studies. And lastly, after the technology transfer to the CMO or the CDMO, ongoing support at the CMO for analytical methods, and also providing any documentation that is required to support our clients' regulatory submissions, and also to provide advice on further scale up, because the scale that you're manufacturing lipid nanoparticle drugs at phase 1 is going to be quite different from the scale that you're going to propose for your commercial launch. And that's very much true of vaccine development. So your initial phase 1 batches for an RNA LNP vaccine will be quite modest. The number of patients in the trial might be less than 50. But by the time you're in phase 3, thinking about commercialization, you're going to have literally thousands of patients in that clinical trial. And the requirement, clinical supply requirement for the drug is going to increase dramatically as well. And you need to get ready for commercial launch because literally, as we all know, hundreds of millions of doses of RNA LNP vaccines have been ordered, have been made to date to basically cover the entire world. And I think we're doing a pretty good job so far, but there's areas of the world that are in desperate need of vaccines for COVID-19. And the pandemic response is something that PNI is also very focused on. And I'll get to that, to tell you a little bit more about that in a couple of minutes. So again, just to recap, we need appropriate analytical tests. They're required to basically determine and define product quality, whether those are in-process tests or final release tests, whether they are tests for your incoming raw materials. They're all required, and it's quite a lot of work. So I just want to remind everyone, it's quite a lot of work to actually develop these tests. It's quite a lot of work to actually perform the studies as well, and PNI can help clients do that. And we can offer the same kind of high quality and rapid development of analytical methods based on our experience and our expertise here at PNI. Not to forget, we're also actively looking at characterization testing, things that might not become release tests, but are also really important for understanding what we're making. Some of these tests may include zeta potential, look at the surface charge on the particle, and cryo-TM analysis and enhanced NMR techniques to look at the ultrastructure, what the inside of these particles actually look like. Are they liposomal? Do they have a dense core? Do they have any interesting features that we want to follow up? It's very important to do that. And particle size doesn't tell you the whole story. So looking at the morphology of these particles is quite important. And we're also always looking at orthogonal approaches for analytics. And in particle sizing, orthogonal methods are also are actively being looked at. So we're constantly evaluating at PNI new technologies that we could actually bring in and help our clients with their drug development needs. So, as kind of alluded to a minute or two ago, pandemic response is something that a lot of people in the industry have been really basically laser focused on in the last year. And PNI is building a facility in Vancouver, our biomanufacturing center, that will actually help with basically pandemic response for the Canadian government. And we've got very grateful for the contribution for the government of Canada. We're going to be offering basically a center of manufacturing excellence for nanomedicine-based RNA vaccines and gene therapies and cell therapies here in Vancouver. And upon its completion in early 2023, planning is well underway. The approximate 40,000 square foot facility will have multiple GMP suites with manufacturing lines for basically the end-to-end manufacturing of genetic medicines. So one of those manufacturing suites will be for RNA, whether it's self-amplifying RNA or messenger RNA, and the other two suites will be for clinical and commercial manufacture of genetic nanomedicines, and most likely, with probably the interest in RNA LNPs, RNA-based LNP manufacturing. So we're very excited to be working on the design and the build-out of this facility, and we feel that we can really make a difference. And with the power of self-amplifying RNA, we are looking at vaccine doses of 1 microgram or less, which is significantly lower than the current doses of mRNA LNP vaccines. So it's going to be a very exciting time for PNI. We are completely focused on meeting our customers' needs. And in this case, the customer is the Canadian government and the people of Canada. And we are, I think very grateful. And I think, I'm very grateful to be part of this industry. I've worked in the drug development industry, working with nanoparticles, basically since my early days after my PhD, working at the UBC in Peter Cullis's and Tom Mann's lab and the spin-out companies from UBC. So, it's been an incredible journey so far for me personally, and I'm very grateful to have the opportunity to work with such a good team at PNI, and we're completely focused on taking this technology forward. We're pretty passionate about it, and I hope in the next few years we can really expand our manufacturing capabilities here in Canada, and also continue to export that technology around the world for basically the greater human good. So I think that's my last slide. I'm certainly happy to take any questions we have now, or if there's a panel at the end of the seminar, you can ask those questions there as well. So yeah, thanks everybody. Thanks for the opportunity.